So in this video, what I'm going to do is take you through the levels that determine the overall structure of the protein. And as I mentioned in the last video, the structure of the protein has a direct determinant of the function of the protein. You think about the different types of proteins that we can have in our bodies, proteins that make structural for structural reasons, so skin, bone, cartilage, hair, versus the proteins that we don't see as much in terms of enzymes, hormones, and so on. Very, very different physically looking, and the reason for that is because their structure really, really links overall with their function. So protein structure is very, very important understanding in terms of understanding biochemistry and how these molecules actually work. Okay, so the first thing we're going to do is go through what's called the primary structure. Okay, so there are 4 levels of structure, and this is our first level. So the primary structure effectively involves the bonding between amino acids. So as I said in the last video, once again, I like to think about proteins or as like kind of a big long necklace, and the amino acids as the beads on a necklace. Okay, so what we're looking at, if we really simplify it, is this concept where we're going to be bonding all of our amino acids all together in a big long chain, effectively. It won't stay as a big long chain, but that's effectively what we're looking at. Okay, so the type of bond and how it occurs. So in this picture here, we've got an example of 2 amino acids. Where the bonding will actually occur will be between the carbonyl group of one amino acid and the amino group of another, only where the primary bonding will occur. The primary bonding will occur between the OH of the carbonyl group and the hydrogen of the amino. Just like always, we don't touch this carbonyl double carbon, too strong, too difficult to break in terms of this sort of bond. You can clearly see your OH and your H, like all of our other reactions, we're going to get H2O cleaved off in this case. All right, so H2O or water is an additional product of this reaction. So the reaction type, or the way in which we name the reaction, can either be called a, I'll talk about the bond first. The bond that is occurring here, that we get to, can easily either be called an amide bond or a peptide bond. Now, I've mentioned amide bond before, and you might remember that, but some of you would also be familiar with a peptide bond, particularly if you study BCE biology. So it really doesn't matter, it's just real personal preference. I'll probably flick between both of them. Okay, so we get an amide bond form between 2 amino acids, between the carboxylic and the amide functional group. And now what we get is what's called a dipeptide. Okay, so you can think about a peptide is literally an amino acid. So 2 amino acids together, or 2 peptides is a dipeptide, and 3 tripeptide. As we get bigger than that, several amino acids joined together, we use the phrase polypeptide, meaning many, and then evidently when we get larger, which most amino acids or most proteins are, as we get to a larger number of amino acids, effectively we use the term protein rather than polypeptide. All right, so some practice just drawing a dipeptide and particularly getting that bonding right, okay? So important here, you can see actually from the diagram, it's given you a little hint because we've got a red OH here and a hydrogen here, all right? So I'm going to draw my first glycine amino acid. Draw my laxative backbone here, NH2H. I've got my carbonyl carbon, and I'm going to leave that bond open. I know my OH is cleaved off and my H is cleaved off to produce water. So now my carbonyl carbon is left open to bond directly to the nitrogen of my alanine molecule, and then I can finish drawing the rest of my amino acid exactly as it is. Okay, this is an example of a dipeptide. Okay, we've just got 2 amino acids joined together. All right, water cleaved off as well. In the next slide, I'm just going to show you how we would look or how we would draw if we have a longer sequence. Now, you're not going to be asked to draw 8, 10, 12 amino acids. Maximum is sort of 3 to 4 amino acids within a sequence. You can see here that in this example, I've given you, or it's shown the basic symbols rather than the full names. That's a really common way of showing a sequence, and then directly shown in terms of an actual drawing here. I really like this initial drawing because this first drawing is actually drawn basically completely correct. And what I mean by that is there's some patterning that actually does occur. So you might notice, but I'll point it out here, is that if I circle all the Z groups, all the R groups, don't confuse you, R groups, here, you can see they're alternating in terms of the diagram. So we do still have a 3D molecule, but what we know with 3D arrangement here in a polypeptide is that the R groups will arrange themselves on opposing poles. Okay, so that's why it's important when we draw them alternating. So if you think about, I suppose this is your plane, your straight line plane, if you want to think about that. And so you'll always have R group down the bottom, R group up the top, R group down the bottom, R group up the top, and so on. The same is also true of your carbonyl carbon. So I might get a different color. I can get a different color. This guy, this guy, this guy, this guy, and this guy. And you can see it also from your amino group. So the idea, and that's because you get a lot of repulsion between particularly that carbon and double bonded oxygen, because you'll get repulsion between neighboring amino acids. So that's the most appropriate way and the most correct way to draw an amino acid. You really don't have to draw the carbon tetrahedral backbone, even though it does exist. In terms of in your exam, it's never really I've ever seen it examined, but I've certainly seen them examine alternating key R groups, particularly if you're asked to draw a particular sequence. The second one diagram down here is more simplified. It actually doesn't have any particular R groups, so it's on that specificity. What I wanted to just have a look at was this diagram down here. Okay, so this is a good example of if you're asked to draw a section of a polypeptide, okay, because you're drawing part of the peptide within a longer chain. What we need to do is our ends need to remain open. So what I'm not going to do, I'm not going to draw NH and H, because that will be my bonds closed, and that means it's at the end of the chain. Because I'm drawing a section, say like it's in the middle of my polypeptide, I need to leave my bonds open, and I usually have my bond and go dot, 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 bond, dot. And that clearly distinguishes that I'm in the middle of my polypeptide chain and not at the start or at the end. That is also definitely examinable, that concept as well. Okay, so the next levels of structure build upon the primary structure. So now what's happening, if you think about your sequence or your big long chain here, what happens is that it starts to become interaction between the amino acids themselves, or between neighboring amino acids, because as we know, all molecules do interact in some form, whether it's a hydrogen bond interaction or a dipole interaction, we'll get that, and that's what starts to create the unique shape of amino acids, and particularly that 3D structure. So where we're going to get to is eventually looking at this structure all the way through. I just really like this diagram here. I can't remember where it's from, but it's kind of a good one, particularly just to have if you create a bit of a summary and want to use some of these diagrams. I quite like particularly that you can see here. Oops, I've just moved that again. You can see the bonding that's actually occurring, okay, in some of these areas. All right, but I'll actually explain what is actually going on first before we look at that. All right, so the next structure is your secondary structure. So what happens as we build our chain is, as I said before, our amino acids do start to interact. And how do they interact? The first level of interaction, secondary structure, occurs due to hydrogen bonding. And it's hydrogen bonding between very specific part of the amino acid. It's between the the oxygen on the carbonyl carbon and the hydrogen of the amino group in neighboring amino acids. Okay, so if you look at the diagram here, you've got an amino acid here and in a amino group, and its interaction between the carbonyl or the oxygen of the carbonyl on a neighboring close by amino acid. So that is always going to be the type of interaction that occurs at the secondary structure. The only difference between alpha and beta is where what kind of amino acids that occurs between. So in an alpha helix, it actually occurs within the same same polypeptide or same chain, and occurs with amino acids that are separated away by 4 units. Okay, so 1, 2, 3, 4 along the chain. Beta-pleated sheets, however, that interaction occurs between 2 different polypeptide chains. So you've got one chain here and you've got one chain here, and there's interaction you can see between the 2 chains. That's why we get 2 different major forms of a secondary structure. Okay, from that point onwards, because we start to really form the overall structure of the protein, we start to form, therefore, the tertiary structure. All right, so it's now folding into a much more complex, much larger molecule. All right, so the tertiary structure effectively delivers the overall 3D shape of the protein and enables the protein to perform its function. The tertiary structure is delivered by, or is determined by the particular. I'll get my back on. The particular R groups present. Okay, so it's the only part of the protein we really haven't touched yet. So we'll just redraw this back up here. I'll just draw a protein completely by itself, not in a chain. Okay, so this is the part we're talking about. We've already interacted this one, and not this one, technically this one, and one of these guys. The hydrogen's not going to really do anything, so the only other part of our amino acid that is yet to form or is yet to really interact in terms of structure is the R group. And so what that means, because the R groups are so varied, basically means we're going to get a lot of varied interactions. And it's important to be aware of that, but also be able to comment on what types of interactions would be present. So this picture gives you a bit of a display of some different kinds. We can have hydrogen bonding. Hydrogen bonding will occur between hydrogen, oxygen, hydrogen, fluorine, hydrogen, nitrogen. So there's going to be lots of different hydrogen bondings that could occur between various R groups. You can definitely have dispersions occur, as we know, between all non-polar R groups. Obviously, slightly weaker bonds, but they will be there. We can have dipole-dipole interactions occurring. We can have what's called a disulfur bridge. So there's 2 amino acids that have sulfur in the R group, and they can interact and form fairly strong covalent sulfur-sulfur bonds within the molecule. And you can also even get some ionic bonds occurring, particularly because our amino acids are generally in solution. And if they're in solution, they're often maybe ionizing depending on the pH. And we can have ions form, so NH3 ions, OH ions, whatever they are. And you can get ionic interaction also occur between different groups. Overall, that type of interaction effectively determines the structure, the overall 3D structure of the molecule. Okay, so if we move on, I've just got you just brought up a picture of insulin here. So insulin is a hormone, and it's a protein. It's a very large protein. You can probably see quite clearly from the colouring as well. We've actually got an interaction between 2 polypeptide chains for insulin, and what I want to show you is a great example to show you the disulfur linking occurring between cysteine amino acids between the 2 chains. There's going to be a lot of other interaction occurring. It's a very, very simplified version of insulin, but it's just an example of I suppose, the type of intermolecular attraction between R groups, that tertiary bonding in action. So this table I'm not really going to go through anymore. It's exactly what I've just said, but it provides a bit of a summary of the type of bonding, and that's what we're really focusing on. You need to be able to explain the type of bonding that occurs at different structures and where it occurs, and using examples would be even better. The fourth level structure, the last bit of this video, is just looking at quaternary structure. So what that means is basically that some of our proteins actually are formed from more than one polypeptide chain. There's quite a number of them that are, but there are some proteins that don't even have a quaternary structure, that are just one peptide chain, and that therefore their highest structure is tertiary. An example of a protein that has a quaternary structure is the one I've given here, which is hemoglobin. You can probably guess from the diagram that hemoglobin has 4 polypeptide chains. They're coloured: the yellow, pink, orange, and purple. That interact at an additional level. We don't have to look at the type of bonding here. It's just you understand that they are the quaternary structure is formed from the interaction between more than one polypeptide chain. So each of these, if you think about it, let's take this pink polypeptide chain. So this has undergone a primary structure, a secondary structure, and a tertiary structure to create its overall 3D structure, which then further interacts with the yellow polypeptide chain and the purple one, and so on. Okay. All right, so that is the end of protein structure. Before we move on to protein reactions.